Between 1347 and 1353, the Black Death killed between 30 and 50% of all Europeans alive. Entire villages emptied in weeks. Cities lost half their populations in a single summer. It was the deadliest event in recorded human history. But the plague did not kill randomly. It selected. And that selection changed the human genome in ways we are still living with today. In 2022, a team from the University of Chicago, McMaster University, and the Institut Pasteur published the first direct evidence of natural selection caused by the Black Death on the human immune system. The study was published in Nature. It analyzed ancient DNA from over 200 individuals who lived before, during, and after the plague swept through London and Denmark. The DNA came from teeth and bones excavated from cemeteries across both countries. Samples spanning a 100-year window centered on the plague years of 1348 and 1349. The question they asked was simple. Did the survivors carry different genes than the dead? Teeth are ideal for ancient DNA extraction because the dense enamel and root structure protect genetic material from degradation over centuries. The researchers extracted and sequenced DNA targeting 300 immune-related genes, comparing the genetic profiles of people who died before the plague to those who survived and died years later. A critical sample came from East Smithfield, a mass burial site used specifically during the Black Death in 1348 and 1349. Everyone buried at East Smithfield died of plague. That meant the researchers could compare plague victims directly against people who lived through it and died of other causes later. The results identified 4 genes where specific variants had shifted dramatically in frequency between pre-plague and post-plague populations. One gene stood out above all others. It was called ERAP2. ERAP2 encodes a protein called endoplasmic reticulum aminopeptidase 2. Its job is to help immune cells recognize and present fragments of invading bacteria to the rest of the immune system. When a macrophage encounters a bacterium, it breaks the bacterium apart. ERAP2 helps chop those bacterial fragments into pieces small enough for other immune cells to detect. People who carried 2 copies of a specific variant, designated RS25-49794, produced full-length functional copies of the ERAP2 protein. Those individuals were roughly 40% more likely to survive the Black Death than people who carried 2 copies of the alternative variant. 40% in a single generation. That is among the strongest selection events. Only a handful of selection events in human history have produced comparable genetic shifts, including the evolution of sickle cell trait in response to malaria and lactose tolerance among dairy farming populations. The people buried in these cemeteries did not know they were participating in one of the largest natural experiments in evolutionary biology. 700 years later, beneath the same city, their bones still carry the evidence of what killed them and what saved the rest. To put that number in perspective, most natural selection events in human evolution produce shifts of a few percent over thousands of years. The Black Death compressed that process into less than a decade. People without the protective variant died in enormous numbers. People with it survived, had children, and passed the gene forward. The team confirmed this was not drift or random sampling error. The same protective variants rose in frequency in both the London and Denmark populations independently. 2 separate populations, 2 separate countries, the same genetic shift in the same direction at the same time. But the research did not stop at identifying which genes changed. The team tested whether those genetic variants actually affected how modern immune cells respond to the plague bacterium. They exposed living human macrophages to Yersinia pestis, the bacterium that causes plague, and measured how efficiently cells with different ERAP2 variants neutralized the pathogen. Macrophages carrying 2 copies of the protective variant were significantly more efficient at killing the bacteria than those without it. This was not just a statistical correlation in ancient bones. It was a measurable biological advantage confirmed in living cells under controlled conditions. But here is where the story takes a turn that nobody anticipated. The same ARAP2 variant that saved lives during the Black Death is today associated with a significantly increased risk of autoimmune diseases. Crohn's disease, rheumatoid arthritis, conditions where the immune system attacks the body's own tissues. The gene that once fought plague now triggers inflammation against itself. Hendrik Poinar, co-lead author and evolutionary geneticist at McMaster University, described the finding as a textbook case of balancing selection. A hyperactive immune system was an enormous advantage during centuries of plague epidemics. But in the modern world, without plague, that same hyperactivity turns inward. Europeans today have significantly higher rates of certain autoimmune conditions compared to populations in East Asia and Sub-Saharan Africa, regions where the Black Death either did not reach or struck with less devastating force. The plague killed up to 50% of Europeans, but had far less impact on populations in China and Africa. Those populations did not undergo the same immune selection pressure. A separate study provided independent support for this finding. Researchers compared the genomes of European Romanians and Roma people who migrated from northern India roughly 1,000 years ago. Despite having completely different ancestral backgrounds, both groups showed the same toll-like receptor gene variants, genes involved in detecting and fighting bacterial infections. Two populations with different origins, living in the same place, evolved the same immune genes in parallel. When the researchers tested immune cells carrying those convergent variants against Yersinia pestis in the laboratory, the cells mounted a distinctly stronger immune response than cells without them. But the 2022 Nature study did not go unchallenged. In 2025, a team from Harvard and other institutions published a response questioning the statistical methods. The critique, led by population geneticist Ian Matheson, argued that the sample sizes were too small and the statistical thresholds too lenient to distinguish genuine selection from random genetic drift. 206 samples split across multiple time periods, and 2 countries leaves very few individuals in each comparison group. Statistical power drops quickly with small sample sizes. The original authors responded with a correction in Nature in January 2025, updating their methodology but maintaining that the core finding held. The debate is ongoing, but the functional evidence, the macrophage experiments, remains uncontested. The McMaster Ancient DNA Center, where much of the extraction work was done, has become one of the leading facilities in the world for recovering genetic material from archaeological remains. Working with ancient DNA requires extraordinary contamination controls. Modern human DNA is everywhere, on skin cells, in breath, on surfaces, and a single contaminating molecule can overwhelm the fragile ancient material. Dedicated ancient DNA labs use positive air pressure, UV sterilization, and strict gowning protocols to prevent contamination. The London samples came from multiple cemeteries, including sites that predate the plague by 3 centuries and sites that postdate it by nearly 200 years. This 100-year window gave the team enough temporal depth to track how gene frequencies changed across the plague event specifically. The targeted sequencing approach examined 300 immune-related genes specifically, not the entire genome. Future studies plan to scale to whole genome analysis. But even within those 300 genes, the team identified 245 variants that shifted frequency during the plague years, far more than expected by chance alone. The plague did not just kill. It reshaped the immune architecture of an entire continent. The waves of plague did not end in 1353. Outbreaks recurred across Europe for the next 400 years, nearly every decade in some regions. Each wave applied additional selection pressure. Each wave favored the same protective variants. Over 4 centuries, the cumulative effect was enormous. Historians have noted that later plague outbreaks killed fewer people proportionally, Cultural changes and quarantine practices explain part of the decline, but genetic adaptation may also have contributed. Yersinia pestis itself evolved alongside its human hosts. Ancient DNA extracted from plague victims across different centuries shows the bacterium acquiring new genetic mutations over time. But the human side of the evolutionary arms race left the deeper mark. The ERAP2 variant that surged during the Black Death is now carried by a majority of Europeans. Every person of European descent carries an immune system shaped in part by a bacterium that ravaged the continent 700 years ago. The genetic echo of the Black Death is not a fossil. It is active, present, and measurable in the blood of living people. The pharmaceutical industry spends billions annually developing treatments for autoimmune conditions that disproportionately affect people of European descent. The question these studies raise is whether that disparity is partly a direct consequence of 14th century natural selection still echoing through modern immune systems. The emerging field of evolutionary medicine argues that understanding the historical pressures on our genes is essential for treating the diseases those genes now cause. And the trade-off is real. The hyperactive immune response that fought plague now contributes to chronic inflammatory conditions that affect millions of Europeans today. The Black Death did not just change history, it changed biology. And we are still paying the price. The team at McMaster and Chicago plans to expand the analysis to the full genome and to additional populations across North Africa and the Middle East. The Black Death did not stop at Europe's borders. It devastated populations across the Middle East and North Africa as well. Those populations may carry their own set of plague-selected variants. Islamic scholars documented the plague extensively. Ibn Khaldun, who lost both his parents to the disease, wrote that civilization itself contracted in proportion to the population loss. Trade routes that once carried silk and spices also carried the plague bacterium, moving it from Central Asia westward along the same Silk Road networks that connected civilizations. The plague arrived in Europe in 1347 aboard Genoese trading ships at the port of Messina in Sicily. Within 3 years, it had reached Scandinavia. The primary vector was the flea Xenopsilla chupis, carried on black rats that infested the grain stores of every trading vessel and warehouse across the Mediterranean. The bacterium spread faster than any army. Entire regions were depopulated within months. Labor shortages reshaped the feudal economy of Europe for generations. But the genetic evidence now tells us the impact was even deeper than the economic and cultural devastation. The plague rewired the human immune system at the molecular level. The genetic distance between pre-plague and post-plague European populations is larger than the distance between many modern ethnic groups, compressed into just 5 years instead of millennia. Beyond ERAP2, the researchers found 3 additional genes with significant frequency shifts, all involved in the body's first line of defense against bacterial invasion. All 4 genes showed changes in the same direction in both London and Denmark, ruling out local genetic drift as the explanation. The 4 genes identified in the 2022 study are not the end of the story. They are the beginning of a new field, pandemic paleogenomics. What other diseases have shaped our DNA in ways we cannot yet detect? Earlier studies had already shown that Yersinia pestis was present in Europe long before the Black Death. Ancient DNA from the Neolithic period, nearly 5,000 years ago, has yielded plague bacteria. The Justinianic plague of the 6th century was the first historically recorded pandemic caused by the same bacterium. It may have killed as many as 50 million people across the Byzantine Empire. Each pandemic left its own genetic signature. The cumulative effect of thousands of years of plague exposure has shaped the immune landscape of Eurasian populations in ways that may take decades more of research to fully map. Modern sequencing technology now makes it possible to read the entire genome of a medieval plague victim from a single tooth. The technology that once required months to sequence a single genome can now process dozens in parallel, opening the door to population-scale, ancient DNA studies that were impossible even a decade ago. The East Smithfield Cemetery, where many of the London samples were excavated, now sits quietly in the shadow of the Tower of London. Most Londoners walk past it without knowing that beneath their feet lie the remains of people whose deaths literally changed the genetic composition of their descendants. Today, a European patient diagnosed with Crohn's disease carries a genetic signature that traces back through 7 centuries to a bacterium that killed half their ancestors. The Black Death is not just medieval history. It is inside every European alive today. And the uncomfortable truth is that the same evolutionary pressure that helped Europeans survive the worst pandemic in history may now be the reason millions of them struggle with chronic inflammatory disease. The plague gave protection. The plague took it back, and the genome remembers everything. If you enjoy evidence-based history, subscribe, because the next episode goes deeper into the pandemics that left permanent marks on the human genome.